So now we're going to be looking at a couple of experiments that have to do with the nucleus. So how proteins that are made out in the cytoplasm get back into the nucleus. So remember, we're talking about proteins such as histones, polymerases, transcription factors, things of that nature. So things that function inside the nucleus, but they're made out in the cytoplasm. So the next two experiments that I'm going to look at are short experiments that illustrate how these proteins get back into the nucleus. So the first experiment here is where we are looking at the signal sequence for the nucleus. So the signal sequence for the nucleus is called the NLS, or the nuclear localization signal. Okay? So proteins that have the NLS obviously end up in the nucleus. That's their ticket. Okay? So with this experiment though, this was the same type of experimental technique that you have to know and that you should know already on what to expect this outcome to be. So remember, if you take the signal sequence off a protein, it doesn't go anywhere. If you add the signal sequence to another protein, then that protein goes to wherever that signal sequence is meant to go to. So in this case, what we did is we took the nuclear localization signal off of a protein that normally goes into the nucleus. We took that signal sequence, the NLS, and we put it on a cytoplasmic protein. The cytoplasmic protein that we decided to choose was the pyruvate kinase. Remember, pyruvate kinase is a cytoplasmic protein that functions in glycolysis. So it lives and works out in the cytoplasm. But what we did is we took the NLS from a protein called the SV40T antigen. Now the SV40T antigen is a viral protein from a virus called the simian virus number 40. And this virus protein, SV40T antigen, when it infects cells, goes into the nucleus because it has an NLS. So what the scientists did then is that they basically took off the NLS from this SV40T antigen protein and they put it on to a pyruvate kinase. So normally, in this first slide here, minus NLS, what we are seeing is that out here in this greenish yellow is where the pyruvate kinase is normally located. It's out here in the cytoplasm. The darker area here in the middle here, this is the nucleus. And as you can see, it's kind of light and dark because it's not full. There's nothing inside here, okay? But now the objective is to take the NLS from the SV40T antigen protein, cut it off of that, and put it onto the pyruvate kinase. Now, when we do that, so plus NLS, now the pyruvate kinase that was originally on the outside here in the cytoplasm, when you add the NLS to the pyruvate kinase, it is now taken up into the nucleus. And now the nucleus is nice, dark, and green. Okay? And now, of course, it's green or yellowish green because, again, this is used by immunofluorescence. So obviously there's a yellow fluorescent protein that was attached to the pyruvate kinase to actually visualize it. But again, the reason that it was able to go from the cytoplasm into the nucleus was by simply adding or attaching an NLS to it. Now in this next experiment that still has to do with how proteins get into the nucleus, this next experiment here is going to demonstrate that not only do you need the NLS on a protein in order to get inside the nucleus, but in certain cases that there are proteins that need a little bit more assistance. And this assistance comes from other cytoplasmic factors. So other proteins and ATP that's found out in the cytoplasm that helps get proteins actually into the nucleus. Now remember, proteins that can go into the nucleus easily are those that are less than 60 kilodaltons, and obviously you have to have an NLS. But those that are greater than 60 kilodaltons in molecular weight and still have an NLS need some assistance. Now again, we're going to be looking at this mechanism in detail coming up. So this is an experiment that shows that there are other factors, cytosolic factors, that are needed to help bring a protein into the nucleus. And of course these proteins still have to have an NLS. So with this experiment here, have your slide. I'm looking at slide 13-34, okay, in topic 25. So what you see up top here is that this is showing the cells, okay. Now what they do here with the cells is that they're going to treat the cell with detergent. Again, remember, the use of detergent is to disrupt the lipid bilayer of the plasma membrane, and in that case you can take things in and out of the cell. And again, you use a low concentration of detergent, because again the idea is not to disrupt the nuclear membrane, but just the lipid bilayer of the plasma membrane. So without digitonin, which is in this case the detergent that they used, so minus digitonin, the membranes and the cells are intact. Now when we add digitonin, we see, if you look closely here in this area, you see a big gap has opened up. So this is where the detergent has disrupted the cell membrane, and we have an opening. And now again, this opening allows us to take things in and out of the cell at will. So now, if you look at the bottom here, which is obviously a big black box, is what it looks like, and then this area here with these little white circles lit up, these little white circles here, as you can see, is the nucleus. And it's white and bright because it's showing that there are proteins that have actually gone and been taken up into the nucleus. So here in the black box, it says something at the bottom called minus lysate. Minus lysate means that what we did is, after we poked holes with the digitonin, we extracted the cytoplasmic factors. So a bunch of cytoplasmic factors and proteins and ATP we took out, and these are called lysate. So we extracted the lysate, and as you can see, well, you can't really see, right, anything happening. It says nothing happened. But now, if we leave the lysate, so all the cytoplasmic components intact, then what we can see is that proteins are able to get into the nucleus. And that is what's shown here and lit up as the nucleus with the proteins inside it. So again, what this ended up demonstrating was that there are other proteins out in the cytoplasm that assist these nuclear proteins. So again, these are nuclear proteins that are greater than 60 kilodaltons, that have to have an NLS, a nuclear localization signal, but then require the assistance of various cytoplasmic proteins. And again, when we look at the mechanism, we're going to see that which cytoplasmic proteins we're talking about that actually help with this process.